So, for part 4, this is especially pertinent for human biology. If you were to go in for a biopsy, they would take a small piece of your tissue, usually a little cylinder. They'd punch it out of whatever tissue they think might be a problem. And that little piece of tissue goes to a lab. Now, you can't just preserve a piece of tissue and slap it on a slide and be able to look at it, because it's even a little piece of tissue is too thick. It's just be a big silhouette. So it needs to be sectioned, sliced into slices thin enough for the light to come through when you look at it through the microscope. But then when you make it that thin, it's hard to make out any detail. So they section it, they mount it on slides, and then they stain it. With stains, you're exposing the tissue in that slice to different dyes, basically. And different parts of a cell, different types of cells will pick up different dyes. Like if you were doing a biopsy and they were worried about cancer, there actually are a couple of stains that will only be taken up by cancerous cells. So later on, a pathologist can look at a slice and go, oh, there's a whole section that's picked up that brown stain. We've got cancer in this breast tissue sample. Incidentally, the people that make these slides work in the labs in the hospitals. My daughter does this. She trained to do this in Cobleskill. There's not a lot of places around where you can train to do that type of technician job. It's pretty decent job, pays pretty decent money, just requires an associate's degree, and there's not a huge number of openings locally because Cobleskill is producing a bunch of them, but other parts of the country, they really are desperate for people who can do this kind of work. So much so that there have been places where they will pay to move you, they will pay you a signing bonus. For places like Colorado, where the nearest place that this training happens is like 300 miles away. So, if you're thinking about a possible career.
Tell them the name of the program.
It's the histotechnician program. Yeah, they're called histotechs. The science of looking at tissues under the microscope is called histology. So, this is what a slice of stained tissue looks like under low power. This is actually a section of pancreas, and it's broken up into bits and pieces. In the cells and tissues lab, you'll have to recognize some of this stuff, even though you won't be looking at a pancreas. These lighter areas are actually where insulin gets made. They're called islets of Langerhans. This area over here, it's got a little bit of pink in it, is actually a blood vessel. That pink is blood. There's another blood vessel. And that's what happens, is that if you train in this stuff, you can look at a slice and know, number one, what it is that you're looking at. Here, casting your own shadow. And here, I'll move it around a little bit. Now, your first question is, looking at a section under low power, what can you see in terms of spaces and layers? And I'm not looking for a description that shows me you know what you're looking at. But just give me a rough idea of what you think you're looking at in terms of spaces and layers. There should be pretty much layers visible here. I'm messing with the focus a little bit. And then you're going to get the same question for middle power. I'm going to go up to middle power here. Here's one of those islets of Langerhans. Here's a very, very fine space that probably wasn't visible at the lower power. There's something weird on with that islet of Langerhans. So this is the thing, you can see more detail at middle power. Even if you've got good eyes, very often you'll see more detail. Like in the islets of Langerhans, you can actually see each one of these dots is a nucleus. Each nucleus is in its own cell. And so you can see there's a bunch of cells in this islets of Langerhans. Now, for the third question, this is by far the toughest question on the lab. And I'm a little worried that we're not going to be able to do it well with this setup. So I'm going to go to high power and see what I can find, because here's the trick. You are looking at cells in tissue. Every nucleus, and the nucleus here are these little rings, these little dotty rings. Each of these is a nucleus. But every single nucleus is in its own cell. And this is actually looking much better than I was hoping it would, because each one of these cells has got a little bitty space around it. And so that shows you where the membrane is. When you look at tissues, a lot of times it's just color and nuclei. And you know you're looking at 500 cells, but it just looks like 500 nuclei with a colored background. This at least, you can actually see this cell with the outline. You can see this cell with the outline. You can see this cell with the outline, this cell with an outline, this cell with its outline. You can see the outlines of the cells that show the edges of them. It's probably because this is epithelial tissue. In the cells and tissues lab, we'll talk about this. This is secretory epithelial tissue. Epithelial tissue, in theory, always faces a space, but often in a gland like a pancreas, you can't see the spaces. You actually can. Been a long time since, I just picked the pancreas at random out of the slide box, and this actually turned out much better than a lot of the slides you would have gotten for this lab. But here's your task for question 3. Draw a little area, label a couple of nuclei. Nuclei is the plural, nucleus is the singular. And also indicate where the cell membranes are, the edges of the cells that separates one cell from another. On this particular setup, it should be pretty easy to do. As I said, some tissues, it's impossible. If you were doing this lab, it would have been a scavenger hunt. You would have been looking around. I just happened to be lucky and settle on this right away. But it really, for me, it's a matter of luck too. Very often you look and you go, I don't see anything but nuclei. Here we can actually see the cells. So that is, this always is a typically short lab, especially for people who know what they're doing on a microscope already. So that is the end of part 4 for the human biology people. And so we will stop it there.